Good morning. I'm John Kramer, and I'm the president of the Hudson River Foundation. And it is my great privilege to once again welcome you to the Foundation's Edward A. Ames Seminar Series. This is the third seminar of 2023. The seminar series is named to honor Ned Ames, a long-standing chair of the Hudson River Foundation's board, much loved by all of us here at the Foundation. Ned's contributions to the foundation are only matched and in fact exceeded by a long career that really had at its core thinking of ways to provide a whole variety of resources and particularly science to the management of important places like the Hudson River and other ecosystems throughout the nation. And I think it's particularly fitting that this seminar is emblematic of not just our much we care about Ned, but also the notion that new science that is of consequence, should be heard about and shared with a wide audience. So we're really happy to have you all participating remotely here with us today. You know, traditionally over the many years that we've had these seminars, we have always had folks here in our foundation offices in New York, and we're looking forward to doing that again when we resume the seminar series in the fall. So stay tuned for that, and where we'll be able to welcome at least some of you back to in-person meetings, and we'll provide a hybrid link as well. So without further ado, I want to turn over the microphone to Jim Lodge, Senior Scientist here at the Foundation, to introduce our speakers for today. Jim.
Good morning, everyone, and thank you again for joining us today. Thrilled to be here with you to invite our speakers, Dr. David Kayzak and Dr. Shannon White. The title of today's seminar is Using Genetic Data to Advance Atlantic Sturgeon Conservation in the Hudson River. So just quickly, a few housekeeping items. We're asking, this seminar will run about an hour, and then we'll have time for questions. So if you can please put questions within the Q&A function, the bullet on the bottom of the tab on the bottom of the Zoom screen. Emma and I will try to organize those, and we'll have Dave and Shannon address as many of those as we can. So I am, as I said, thrilled to have you guys here, and I'm really excited for today's talk. This project is a really great example of a research project that has a clear line to informing management decisions. There's a phrase that John likes to use that I'm stealing today is it's research that is use-inspired, useful, and used. And that last one is being used is probably the most challenging. And Dave and Shannon's project, at least components of this project, was conducted in partnership with Rich Pendleton and Amanda Higgs from Cornell, and the DEC Hudson River Fisheries team, led by Greg Kennedy at the New York State DEC's office in New Paltz. And Greg, Rich, Amanda, and all of Greg's team are longtime collaborators of the foundation. So again, I'm really looking forward to this talk and following the research and the management discussions that follow. I'd like to first introduce Dr. David Kayzak. He's a supervisory research field biologist with the USGS Eastern Ecological Science Center, where he leads the conservation genetics and genomics research team. And Dr. Shannon White, also a fisheries ecologist with the USGS Eastern Ecological Science Center in West Virginia. So Shannon and Dave's work combines molecular techniques, novel field methods, and advanced statistical modeling to provide decision-relevant information for species of conservation concern. Please join me in welcoming Dave and Shannon.
Thanks, Jim, for that introduction. I think you hopefully can see my screen now, but certainly stop me if you can't. So Dave and I are really excited to be here today to share some of the research that we've been doing really the last couple years to advance sturgeon conservation, specifically in the Hudson River. And we're going to jump all around throughout this talk, but just keep in mind that the umbrella theme is, how we're really using genetics as a tool to advance our understanding of sturgeon ecology and conservation. So before we really get started though, Dave and I, we are two members of a larger research team at USGS. And really without the efforts of the other conservation geneticists that we work with, including Barb Lubinsky, Robin Johnson, Mike Eccles, and Maluska Oliveira Hyde, the research we're talking about today absolutely would not have been possible. So definitely a special shout out to them. But also we at USGS are really part of a much larger collaborative network of folks that are located from Georgia all throughout Maine and even into Canada. And as we're going to talk about today, sturgeon conservation is by nature a collaborative teamwork effort. And without these folks really buying in to sharing their data and working together to conserve Atlantic sturgeon, we certainly would be much further behind than where we're at today. And since we are going to talk today about the Hudson River population, I do want to take a moment to give a special thanks to our collaborators at New York State DEC, Delaware State University, and the University of Delaware, who have been really instrumental in all of the research projects we're talking about. But really, basically, all of the work that's going on in the Hudson River is generally being supported by folks at those three universities and institutions. So diving on in here, let's introduce the star of today's show, the Atlantic sturgeon. For those of you who haven't had the pleasure of seeing this fish up close and personal, I really don't think I could oversell just how big this fish is. Adults can reach upwards of 800 pounds and grow as long as 14 feet. And so to put that into perspective, at 14 feet, that makes an adult Atlantic sturgeon just a little bit shorter than the average giraffe. So this is an absolutely massive fish. Now, some of you may have heard Atlantic sturgeon being referred to as kind of a living fossil, and that's because Atlantic sturgeon were swimming around our rivers and estuaries at the same time that dinosaurs were roaming the earth. So Atlantic sturgeon and most sturgeon species are some of the oldest fish that are still alive today, and in fact, they're some of the oldest animals or taxa in general that are still existing on our planet. Atlantic sturgeon also have some pretty unique life history characteristics, including the fact that they are anadromous. So you might have heard this term in reference to Pacific salmon, where an individual is born in a freshwater river. They then migrate out to the ocean where they spend most of their adult life and then return to their natal freshwater river to spawn. Now, unlike those Pacific salmon, though, Atlantic sturgeon are idiroparous, meaning that they can reproduce multiple times throughout their life cycle. So they will complete that freshwater to saltwater migration many, many times throughout their entire lifespan. Speaking of that lifespan, Atlantic sturgeon can live up to 60 years old, but it does take between 10 and upwards of 30 years old for an Atlantic sturgeon to reach sexual maturity. So when we're talking about this fish, it's definitely a species that's living in the slow lane, so to speak, where we're measuring a lot of these life history events on the magnitude of several decades at a time. Now, Atlantic sturgeon, they've been in the news a lot recently, but human interactions with this species really date back to hundreds of years ago when Atlantic sturgeon were supporting subsistence fisheries for indigenous peoples, as well as early settlers to America. So quite famously, our early colonists were pretty bad at farming for themselves and quite literally almost starved to death. But once they figured out that Atlantic sturgeon were swimming in their backyards, they were able to harvest this species and actually sustain themselves. And so Atlantic sturgeon are literally credited with saving some of our colonies like Jamestown. And judging from this quote from John Smith in the early 1600s, you can see that there was no limit to the amount of sturgeon that were available for feeding both humans, as well as even pets and probably even livestock. However, things really did start to change in the 1800s. At this time, there was really a shift away from that subsistence fishery and more into a focus on commercial harvest, and specifically commercial harvest to support caviar demands. So based on this graph that I have here on the right, that graph has date on the x-axis and removal of Atlantic sturgeon in tons on the y-axis. And you can see in the late 1800s, we kind of had peak Atlantic sturgeon harvest at about 3,000 tons per year. But within about 10 years, Atlantic sturgeon harvest really began to plummet. And that is because within the span of about 10 years, most Atlantic sturgeon populations would be harvested to depletion. So by the turn of the 20th century here, a lot of our populations are already starting to collapse. And by this point, a lot of populations probably are already functionally extinct and on their way to extirpation. Now, if we look at that inset though of that graph, we can see that Atlantic sturgeon harvest did continue throughout the 1990s, albeit at very severely reduced landings. But in 1998, there was a bit of a monumental decision in the decision to close the Atlantic sturgeon fishery. So in 1998, there was an Atlantic sturgeon harvest moratorium issued. So we see that this graph starts to transition away from reporting commercial harvest and into commercial or dead bycatch instead. So I want to point this out because even after we've stopped Atlantic sturgeon commercial harvest, we're still getting about 5 to 10 tons of Atlantic sturgeon dead each year, just caught up in bycatch. And this becomes symptomatic of the fact that pressures to Atlantic sturgeon are not just isolated to harvest issues. And really, as times have evolved, what we're seeing is that increasingly Atlantic sturgeon are being threatened by other anthropogenic activities as well. So in addition to fisheries bycatch, things like habitat loss, as well as incidental mortality through things like ship strikes and marine construction, continue to slow harvest recovery. And I think it's pretty clear that in some populations, these pressures are also actually contributing to ongoing population declines. So in recognition that that harvest moratorium was really not enough to protect Atlantic sturgeon were listed under the Endangered Species Act in 2012. As part of this listing, populations were divided into 5 distinct population segments, or as we'll refer to throughout the rest of this talk as just DPSs. A DPS is meant to represent kind of a collection of rivers that are genetically and physiologically similar. And as of today, there are currently 18 recognized genetically distinct populations. And it's important to note that we are actually literally working every day to increase that number and to hopefully characterize and describe even more populations of Atlantic sturgeon. Now you're going to see this graph on the left throughout the rest of this talk kind of spring up here and there. And so I want to take a second just to kind of orient you to what this graph is showing. You can see there, it's kind of highlighting the distribution of those 18 spawning populations. And you can see there in the south, some rivers actually contain genetically distinct spring and fall runs. So again, they're genetically distinct populations, but they're kind of 2 different life history strategies there. These rivers are color-coded by which DPS they belong to. And in 2012, when Atlantic sturgeon were listed under the ESA, the 4 southernmost DPSs were listed as endangered, whereas the Gulf of Maine was listed as threatened, due largely to limited perceived threats and probably also quite a bit of limited data as well. Now, the 2 Canadian rivers up there, they are outside of the purview of the ESA, but they do have also special conservation protection administered in Canada. Now, in terms of application of genetics, we kind of took a pretty big step forward a couple years ago when myself, Dave, and a bunch of co-authors published the microsatellite genetic baseline for Atlantic sturgeon populations in the entire North America coastline. Within this baseline manuscript, we described kind of the distribution of genetic diversity and differentiation of those 18 populations, which is all well and good in its own right. But the reason that this baseline has been so important for us is it's really increased our ability to use genetic assignment tests in Atlantic sturgeon conservation. Now, for those of you who are maybe not familiar, a genetic assignment test is a statistical tool where we use allele frequencies to determine the population origin for an unknown fish. So to put this a different way, if you hand me a small tissue sample from an Atlantic sturgeon, we can use our genetic baseline and perform an assignment test and tell you where that sturgeon was likely born. And this might seem like kind of a mundane or silly research objective, but it turns out knowing the natal origin of an Atlantic sturgeon is the key to unlocking a lot of information about basic sturgeon ecology, as well as predicting threats to survival. And so for the remainder of our talk, we're going to kind of discuss some of the research that we've been doing under this umbrella framework. And to kick that section of this talk off, I'm going to hand the presentation over to Dave.
Thanks, Shannon. So now that you have a background on Atlantic sturgeon, I want to share with you some of the ways that our team is using genetics to help improve our understanding of the species and in turn support their conservation. Our efforts include trying to better understand the impacts of historic fisheries, estimating abundance so that we can evaluate the status and trends for populations, and identifying critical habitats and tracking sturgeon movement across the broad array of habitats that they use. So for the remainder of the talk, we'll highlight some of the projects that we've completed and what we've learned about Atlantic sturgeon. So as Shannon described earlier, historical overfishing is one of the major causes that led to population crashes and ultimately listing under the Endangered Species Act. Because sturgeon are a really long-lived fish with late maturity and low rates of natural mortality, we may still see the legacy of past fisheries in today's populations. And a better understanding of the status of sturgeon populations during the final years of commercial fishing might help us understand the impacts of those fisheries and how things have changed in the last few decades. We know that in the final years of the commercial fisheries, that landings were rapidly declining, which in turn suggests that the populations that supported those fisheries were also rapidly declining. One of the last Atlantic sturgeon fisheries in the US operated in the New York Bight, pretty close to the mouth of the Hudson River. This fishery was relatively small in comparison to some of the heyday fisheries of the late 1800s, but potentially still quite significant given the depleted state of populations at the time. And it's also noteworthy that this fishery continued to operate after many other states closed down their local fisheries. In general, my impression is that these fish were assumed to be from the Hudson River population. And in fact, I've heard it referred to as the Hudson River intercept fishery. But at the time, we really didn't have a way to test that assumption. But if we fast forward a few decades, now we have powerful genetic tools that allow us to assess the natal origin of a sturgeon from just a tiny sample of DNA. One of our colleagues, Dr. Dave Secor at the Chesapeake Biological Lab, had collected fin spines from this sturgeon fishery in the 1990s to look at age and growth, basically using a similar approach to how you count rings on trees or scales. And those samples, which a few of which are shown on the screen right now, had been archived after they were done with them and basically just sitting in a closet for about 25 years. But we realized that they likely still contained enough DNA to go back and test the stock composition of sturgeon that were harvested prior to this coastwide moratorium. So after some tinkering in the lab to figure out how we could best extract DNA from a dried up bony structure that was glued to a piece of glass, we were able to extract enough DNA to genotype many of those historical samples. And what we found was that indeed, most of the sturgeon harvested in the final years of the Hudson River intercept fishery were from populations in the New York Bight, predominantly from the Hudson River, but there were also contributions from the Delaware River. And that's not really too surprising given the proximity of this fishery to those rivers, the Hudson and Delaware Rivers. But there's actually more to the story. In addition to sturgeon from the Hudson and Delaware Rivers, we also see quite a few sturgeon from other rivers across the coast, including individuals from all 5 distinct population segments and also Canada. So rather than just impacting local populations, the fishery actually exploited populations from across a much broader area. And that's a theme that we're going to keep coming back to. Atlantic sturgeon can range over broad areas, and local decisions can impact populations at much broader spatial scales. So moving forward, now that all fisheries within the United States for Atlantic sturgeon have been closed for several decades, how are our populations doing? We can answer that question from both a genetics perspective and also from a demographic perspective. In other words, how many sturgeon are currently out there, and do they have enough genetic diversity left to thrive in the future? And I'm going to start with the genetics perspective. When we look at patterns of diversity across the range of Atlantic sturgeon, from the Satilla River all the way down south in Georgia, up to the St. Lawrence River in Canada, we see what I would consider to be a decent amount of genetic diversity within populations. There's nothing here that really strikes me as a cause for concern in comparison to the many other imperiled species that we've worked on. we see that genetic diversity is relatively similar across all of our baseline populations, and also that the Hudson River population is on the higher end of the range of what we observed. All of that's, I think, very encouraging. There's nothing outwardly to suggest that there are genetic problems, as often occur when we have populations that crash and go through a prolonged bottleneck. And I think for Atlantic sturgeon, some of those same life history characteristics that make them difficult to recover also likely help them retain genetic diversity. They live a very long time, and they have many overlapping generations, both of which help to reduce the rate at which diversity is lost by chance in small populations. It's also worth mentioning that part of the genome of Atlantic sturgeon is tetraploid, meaning that there are 4 copies of each gene. And that should help further buffer populations against losses in diversity that are associated with bottlenecks. Okay, so now let's shift to the demographic perspective. How many fish do we have? As we work to conserve and manage Atlantic sturgeon, it's really important that we understand the status of our populations and whether abundance is increasing, decreasing, or staying the same. That's key information that helps us understand if our conservation actions are working or if we need to make changes. That said, unfortunately, Atlantic sturgeon are, even as fishes go, particularly difficult to count for a variety of reasons. Since the closure of all US Atlantic sturgeon fisheries in the 1990s, we don't have any commercial fisheries landings data to use to draw inferences. Atlantic sturgeon have coastwide migrations, and they can travel over broad areas. They have extensive mixing of stocks in estuarine and marine waters. Shannon will talk more about that in a few minutes. They don't spawn every year. The interval between spawning efforts varies among individuals, all of which means they're never all in one place to count. And for the Hudson River population in particular, there are multiple potential spawning sites. The river is deep, often 40 to 60 feet wide and 40 to 60 feet deep and a mile wide, excuse me. There are quite strong tides, there's frequent shipping traffic. So, we have our work cut out for us. And given all of these challenges, what my team has been doing is working to develop new, more tractable methods that allow us to estimate abundance and better understand the status of our populations. So next, I want to share an approach we developed to estimate the size of the adult spawning run in the Hudson River. And rather than focusing on what wouldn't work and all the challenges, we took the opposite approach and started mapping out what data sources were actually available for this population. We knew that some of the returning adults were fitted with acoustic telemetry tags, and those send out unique coded signals every few minutes. We knew that there was a network of hydrophones along the Hudson River which recorded those signals. So we were able to tell things like which tagged individuals entered the river, when they were there, and how far upstream they moved. And all that information is great, but in isolation, those records only tell us about the tagged fish rather than the total population. That is really what we care about. So we needed a way to relate the number of tagged fish to the total population. And as it happens, at the time, there was a graduate student who was using side-scan sonar to count the total number of fish in the Hyde Park Reach, where we were also recording acoustic tags. So we now have this secondary data source that allows us to relate the total number of sturgeon in a specific reach with a number of tagged sturgeon in that same reach. And we worked out a mathematical model that allows us to leverage those data sources and estimate the annual spawning run size. So let's talk through that approach in a bit more detail. As I mentioned before, there's a network of hydrophones on the Hudson River that detect tagged fish. On the map here on the right, each circle represents one of those hydrophones. The network goes all the way from the mouth of the Hudson River all the way up to Albany. And we can use the hydrophone network to track the upstream migration of each tagged fish and count how many total tagged individuals moved upstream. In this case, based on our understanding of the Hudson River and its sturgeon, we assumed that sturgeon moving upstream of Bear Mountain, those are the hydrophones that are now shown in gray, that they were on a spawning migration. So we have a count of the total number of tagged sturgeon moving upstream in a given year. We then use side-scan sonar to count the total number, that is both tagged and untagged individuals, of Atlantic sturgeon in the Hyde Park Reach on 12 days that were spread across the spawning season. And that Hyde Park Reach is a relatively small portion of the overall river, which is currently shown on the receivers in red on the screen. And it's an area where many sturgeon are known to aggregate during the spawning season, and that's likely due to the presence of rocky spawning habitat. Those sonar surveys basically entail towing the sonar unit down a series of parallel transects, and then later counting the number of sturgeon targets in the imagery. I'm currently sharing an example of a sturgeon target in the sonar imagery, and basically we see a bright return that comes back from the fish itself. The sturgeon are quite bony and reflect sound waves pretty well. And then beyond the bright target that's the fish itself, we actually see a shadow in the imagery where we can make out the profile of the fish, including its snout and the distinctive heterocircle tail. And as those transects are being completed, we tow a hydrophone to listen for the tag sturgeon in the same reach. So how do we put this all together? The math here, the actual math is pretty complicated, but the general principle is pretty straightforward. So as a hypothetical example, if we heard 10 tag sturgeon in the Hyde Park Reach, and we identify 100 sturgeon targets in the sonar imagery in that same geographic area, we can estimate that 10% of the spawning run is tagged. In this example, if we had 150 tagged adults that were detected moving upstream on a spawning migration, we could then extrapolate to an estimated spawning run of about 1,500 adults. And by applying that same logic to real-world data from the Hudson River, we estimated that the 2014 spawning run for Atlantic sturgeon consisted of about 466 individuals with a 95% credible interval between 310 and 745 animals. If we put that in context, this suggests that the Hudson River probably supports one of the larger populations of Atlantic sturgeon. Although we need population estimates from other rivers to be able to say this with more confidence. But even though it's currently one of our larger populations, the spawning run size for the Hudson River is still almost certainly much, much smaller than it was prior to exploitation, and doesn't appear to have grown much since the closure of commercial fisheries. Our estimates from 2014, but I will mention that some of our colleagues from New York DC and University of Delaware, Delaware State University are currently applying the same approach to generate new estimates. In fact, there's a good chance they're out on the river today. So at this point, I'd like to pass the microphone back to Shannon, who's going to pivot and talk about juveniles in the Hudson River.
Thanks, Dave. So as Dave was just going through our spawning run size estimation, I hope you gathered that it's critically important that we estimate adult run sizes, but at the same time, it is super difficult, right? It requires a ton of data and a lot of really complex statistical methods. And so realistically, it's probably not going to be possible for us to estimate spawning run size every year. And so because of that, we want to kind of shift our attention and see if there's other demographic data that we can use to monitor population recovery over time. And one focus of our attention lately has been juvenile population sizes. So unlike adults, juvenile and subadult Atlantic sturgeon can be a little bit easier to capture because they tend to overwinter in very discrete areas of habitat. And so recognizing this, New York State DEC has actually been surveying juvenile and subadult Atlantic sturgeon since 2004. And from this survey data, they estimate an index of abundance and then use that estimate to track recovery over time. Now, this is all well and good, and it's super informative, but there's always been one small hiccup to this survey data, and that's that it is kind of unclear whether individuals that are overwintering in these habitats are only natal to the Hudson River. And specifically, we don't know a ton about juvenile and subadult movement. And so there was always the potential that individuals from outside sources could have migrated into the Hudson River. And if that is the case, it would suggest that that survey data might be biased by the presence of those non-natal fish. So understanding natal origin of juvenile and subadult Atlantic sturgeon was actually the topic of the HRF research grant that Jim mentioned at the beginning of this talk. This project started in 2021 and we just wrapped it up a couple months ago. As part of this project, we genotyped and sexed over 500 juvenile and subadult Atlantic sturgeon that were captured during those annual surveys in Haverstraw Bay. All of these individuals were captured somewhat recently between 2017 and 2022. We then used that genotype data to determine the natal origin of all of those 500 individuals. And going into this study, we did kind of hypothesize that larger fish should probably be a higher likelihood of being non-natal. So to understand how fish age potentially influence natal origin, as Dave mentioned earlier, we took a small fin spine or a section of the fin ray from most of the individuals in this study and determined age by counting the rings on these hard parts. So to show you kind of what that looks like, this is an image of one of those fin spines for an individual that was estimated to be about 5 years old. So big picture with this study, what we wanted to do is first of all, determine what proportion of individuals were non-natal in our survey data. And then at the same time, we wanted to see if potentially age influenced natal origin, with the understanding that if it does, maybe we can just kind of adjust our survey methods in order to better document and represent that Hudson River population. But if we look at our results, somewhat fortunately, what we find is that almost all of the fish in the survey are in fact natal to the Hudson River. So this graph here on the left shows you a total length of a fish on the x-axis with the likelihood that that fish is natal to the Hudson River on the y-axis. And each of those red dots represents an individual that assigned with highest likelihood to the Hudson River. So over 88% of the fish in our survey do appear to be natal to the Hudson River. Now, there were 57 fish that assigned to a river that was not the Hudson, but after we ran our numbers and looked at our statistics, what it turns out is it doesn't seem to be kind of any predictive ability in terms of size, age, or sex in determining that the likelihood of a fish is natal or non-natal to the Hudson River. And this is important because it does suggest that a non-natal fish just kind of randomly occurs in our survey. So there's no way to kind of adjust our survey methods to avoid them. But in general, it doesn't really matter because most of the fish in our survey are natal to the Hudson River, meaning that these survey data do seem to do a really great job of capturing trends in juvenile and subadult abundance of that Hudson River population. And so zooming back out, now that we know that the juvenile indices of abundance are monitoring the Hudson River, and we can estimate spawning run size, we now have the ability to really monitor demographic trajectories throughout most stages of an Atlantic sturgeon's life cycle. And specifically, the juvenile abundance indices, they can provide kind of an early look at what population trajectories are looking like and what adult populations should look like in 10 to 20 years. And then by estimating that spawning run size, we can see if we're actually effective at meeting some of our recovery targets and goals. And if we're not, because we're now able to monitor populations at multiple life stages, it starts to improve our ability to really target which life history or which life cycle might be the biggest bottleneck to recovery. One thing I didn't really mention in a couple other slides is that one additional tool that we've recently added to our toolbox is the ability to estimate or determine individual sex based on non-lethal genetic methods. And this is turning out to be really important and really informative because in our juvenile survey, we had basically a 50-50 split of males and females, as one would expect from a natural and healthy juvenile population. But what we're finding is that adults that are returning to the Hudson River to spawn are predominantly male and quite clearly biased towards males. And so this could suggest that there is some sort of female-specific mortality event that's occurring either in the lower estuary or more likely probably in the ocean. But right now, all of that is kind of hypothetical and certainly would be a future line of research down the road, though. So I want to transition now away from estimating demographic and population sizes and really talk now about habitat use, starting with habitat use specifically in the Hudson River. So just as a quick refresher here, as an anadromous species, Atlantic sturgeon really don't spend too much time in these freshwater rivers. They spend most of their adult lives in the ocean and then return to the Hudson River kind of in late May or early June, spend maybe a couple days up to a few weeks in the Hudson River before returning to the ocean, where they spend a year or three before repeating this cycle all over again. In addition, it's important to point out, as Dave kind of mentioned, Atlantic sturgeon really require specific habitats in order to spawn. So there's miles and miles of undammed river in the Hudson River, but we're really only talking about a couple key areas of the river that are important for the spawning population. So given that that spawning window is really a pretty small window, and we're really talking about only a couple 100 yards of habitat, it's obviously very important that we have a firm understanding of the timing and location of spawning in order to make sure we're protecting that next generation of Atlantic sturgeon. So to get at this topic of in-river habitat use, we're going to return to that acoustic telemetry database that Dave mentioned a little bit ago. And specifically, as we're talking about spawning habitat, I want to again mention that we're of course interested in that Hyde Park reach, since that is the primary area of Atlantic sturgeon spawning. But I am going to mention that there is kind of a secondary unconfirmed area of spawning at Catskill that we also kind of monitor Atlantic sturgeon habitat use, because it does seem to be important, even though we haven't 100% definitively documented spawning there. Now, Dave looked at a kind of a limited subset of the data that is available to us right now. But for this analysis, we really incorporated data for a lot of Atlantic sturgeon. In fact, over 300 Atlantic sturgeon that were tagged off the coast of Delaware. Importantly, not all of these fish are natal to the Hudson River. In fact, only about a third of them have been detected so far in the Hudson River. But that 100 or so Atlantic sturgeon have amassed over 14 million detections in the last decade. And so 14 million, that's obviously a huge database, but importantly, it's really taken this long to generate enough data to really tease apart individual variation, as well as variation that might be occurring due to sex or natal origin. So with over a decade of data, it's very, very hard to really distill these patterns down to just a couple slides. But instead of throwing a bunch of really complex statistics and graphs at you, what I wanted to do is actually kind of show you some animations of some representative habitat use for a couple of different groups of fish. So before I start these animations, though, I kind of want to take a second just to orient you to what you're going to be looking at. When I start these animations, they're going to show habitat use of an adult male on the left and an adult female on the right. And both of these fish are natal to the Hudson River. And so you can see there a red dot for the male and a green dot for the female. And as I start this animation, I want you to kind of pay attention to how these dots are moving around the river, but in particular using habitat at Hyde Park and Catskill. And you can see pretty clearly that that adult male quite quickly moves upstream, spends a couple days in Hyde Park, and actually leaves Hyde Park before the female even gets there. The female almost immediately turns right back around and leaves the river on June 4th. Whereas that adult male continues to occupy habitat upstream of Catskill before eventually kind of migrating back down, loiters around Bear Mountain, and then eventually does leave the river at the end of June. And so again, these are just kind of representative patterns of two individuals, two of really 100. But if we looked at all of our data, what we are seeing is a pretty consistent pattern where males do tend to spend a lot more time in the river, also tending to move further upstream and also move kind of up and down the river much more frequently than do females. I'm going to show you that same adult male that's natal to the Hudson River on the left, but this time I'm going to compare it to a non-natal male on the right. This non-natal male enters the river a few weeks later, but you can see that male almost immediately hightails it up to Hyde Park, where it kind of hangs around that spawning habitat. And what we're going to see is that even after that natal male migrates back down into the ocean, that non-natal male is going to continue to occupy habitats in the Hudson River. And in fact, I think that this non-natal male actually ends up spending over 2 weeks longer in the Hudson River than does the natal male. And again, representative patterns. but pretty consistently, this is what we're seeing in our data. So just to, again, try to summarize this really complex data set here, what we're finding is that generally speaking, males do, again, seem to move much further upstream compared to females, and move among habitat reaches much more frequently. They occupy that Hyde Park reach for a little while, but continue to kind of leave and come back multiple times throughout the year. In terms of timing of habitat use, though, females, like I said, do spend a lot less time in the river. But what's interesting is that if we just kind of focus in on time spent at Hyde Park, there's really not a lot of overlap between males and females in that spawning reach. I think across the entire 10 years of data, I've been lucky to find, less than 5 days or so where there seem to be tagged females and males both within Hyde Park. And then something that we're really scratching our heads about right now is the fact that we are consistently detecting non-natal fish in the Hudson River during spawning season. And this is a bit of a curiosity because everything that we know about Atlantic sturgeon ecology would suggest that adult fish should really only return to their natal rivers when it's time to spawn. And so the fact that we're detecting non-natal fish is a little bit interesting and a little bit of a conundrum right now. What these non-natal fish are doing in these river systems, we're not quite sure. We don't think that they're spawning based on genetic evidence. But again, what they're doing outside of potential spawning, best guess is in the eye of the beholder right now. But regardless of why they're there, I think that this really does point to the fact that the Hudson River is just generally an important habitat for not only the Hudson River population of Atlantic sturgeon, but also sturgeon from all the way down in the southeast as well. So that's habitat use in the Hudson River. What's going on once these fish are in the ocean? So big picture, they move around. They move around a lot. Studies have shown that Atlantic sturgeon can move over 800 miles once they're in the Atlantic Ocean, really highlighting the fact that these fish are potentially exposed to threats across really vast spatial scales. In addition, if you remember, because Atlantic sturgeon can live so long, they might spend upwards of 20, 30, or even 40 years in the ocean. So these habitat threats can really be realized across entire careers of fisheries biologists, really underscoring the need for collaborative studies of marine habitat use in order to really piece together how fish are using different environments in the open ocean. And so understanding marine habitat use is something that we've been working on through a lot of different types of studies, one of which is a genetic mixed stock analysis, where basically Atlantic sturgeon that are encountered in the river or in the ocean, a small fin clip is taken and sent to us at USGS, and then we run one of those assignment tests in order to determine where a fish was actually born. So this graph here on the right is showing you the results of one of these mixed stock analyses that was completed in 2021. The orange squares correspond to fish that assigned to either the Delaware or the Hudson River. And I can tell you that by and large, most of those orange squares do correspond to a fish that's natal to the Hudson. So if you just look for these orange squares, you see that fish from the Hudson River do kind of routinely show up as far north as Long Island, New York, but also as far south as Cape Hatteras, North Carolina. So Atlantic sturgeon from the Hudson are moving around. They're getting out throughout the entire mid-Atlantic region there. But an important thing about these mixed stock analyses is that really they only represent where a fish is located at a single point in time. It doesn't really tell us much about how fish are moving through all these in different environments and potentially how they're entering into other non-natal river systems. And so to address this question, us at USGS have formed a collaborative network with 24 other institutions to look at coast-wide migration patterns of fish throughout the entire coast. So this coast-wide database so far has amassed over 32 million detections for Atlantic sturgeon, with each of those dots on the map that you see there representing one of those acoustic telemetry receivers that's constantly recording data for us. This large database has so far Been tracking the movement patterns for just under 2,700 Atlantic sturgeon, of which over 600 of them did assign to the Hudson River. And so this project is still ongoing, but we do hope that by the end of it, we can get a much better understanding of individual habitat use, as well as population and DPS specific patterns of migration. And again, with just over 600 fish from the Hudson River, I think it's going to provide a much better understanding as to how fish from this population are using marine environments. And then the final stop on this kind of tour of research activities of ours is a project that Dave and I just started a couple months ago, where we're going to actually use satellite tags to provide kind of a continuous monitoring of Atlantic sturgeon movement and habitat use in the ocean. And the purpose of this study is really to see how Atlantic sturgeon are potentially encountering areas of human disturbance. So this could be areas of wind energy development, but we're specifically focused on how Atlantic sturgeon are potentially using areas that are being dredged for sand. So we have a study area that's just off the coast of Virginia Beach that is kind of an active dredge site. And so we're going to be using a combination of the telemetry and side scan sonar to see how these fish are using those habitats, and potentially how this dredging disturbs their marine environments and their occupancy and distribution. Because this area that we're talking about is kind of just off the coast of Virginia Beach, which is not too far from the Hudson, we do think that over the course of the study, we are going to recruit quite a few Hudson River fish into our analysis. But again, we are just at the very beginning stages of this project, and it is meant to go for about 4 years. So certainly stay tuned for those results. And so we're going to kind of start wrapping things up now. And to do that, I'm going to hand this talk back over to Dave.
All right, thank you, Shannon. So to wrap things up, in conclusion, we've leveraged genetics and a whole suite of collaborative partnerships to learn a lot more about Atlantic sturgeon and how human activities may be impacting their recovery. In the Hudson River, our estimates suggest that the adult population size is much smaller than it once was, but it's still likely one of the larger contemporary populations of the species. Our estimates also suggest that the population's been relatively stable since the closure of commercial fisheries in the 1990s. When we look at juvenile sturgeon in the Hudson River, they're overwhelmingly from the local spawning population, but that story changes as we look at larger individuals. Our work has really reinforced that individuals frequently travel far from their natal rivers, making it important to consider potential threats, not just in the local area, but over much, much broader spatial scales. We discovered that adult Atlantic sturgeon from other populations are also using the Hudson River, not just the lower estuary, but they're in some cases moving quite far into upstream habitats. Incidentally, I don't think we mentioned it today, but we're also seeing sturgeon from the Hudson River move fairly far upstream in other rivers, all of which changes our understanding of when and where each population occurs, and in turn, how we might best monitor populations. We've learned that adult males and females use riverine habitats differently, with females making much shorter forays to key spawning habitats. And overall, collectively, we're learning a lot about sturgeon, their status, and what's impacting their recovery. And we hope that all of that information helps to improve conservation outcomes. We're seeing some positive signs, but there's still serious threats that are ongoing, and there are other new threats on the horizon. Ultimately, our science can help enable conservation, but the decisions that society more broadly makes in the years ahead are likely to have a big impact on the future of sturgeon. These are fish which have been around since the time of dinosaurs. They've survived meteor impacts, ice ages, and countless other changes. But today, most wild sturgeon populations around the world are really struggling to coexist with people. We're lucky in that the Hudson River population of Atlantic sturgeon seems to be doing better than most, but even it's still quite vulnerable. But I want to end on something a little bit more positive. A few years ago, some of my colleagues were out on the Hudson River doing side scan sonar surveys, and they imaged a sturgeon that was just over 4 meters in length. We knew historically that sturgeon could reach this size, but no one had actually observed an Atlantic sturgeon this big for quite a while. We don't really have a good idea of how old a fish that size might be, but I think it's really safe to assume that this is a very old fish, much older than me. And frankly, I think it's amazing that we still have fish out there that survived the intense commercial fisheries of the past, that have likely logged tens of thousands of miles swimming up and down the Atlantic coast. That's a fish that's undoubtedly seen a lot and survived some really significant changes in the world. And that really highlights to me that we have a lot left to discover in the marine environment. There are still some really cool things out there. And giant prehistoric fish like sturgeon that return to the Hudson River annually are some of the natural wonders that, in my opinion, make our world a really interesting place. So on that note, I want to thank you all for taking time this morning to tune in and learn more about sturgeon. And I believe we have an opportunity for some questions.
Yeah, well, thank you, Dave and Shannon. That was a fantastic presentation. On behalf of everyone, all the participants, let me just thank you for participating today. We do have a bunch of questions. We will start with some sturgeon ecology questions. Helena and Draco, in my office here at the foundation, she asked if males and females are not spending much time together in the spawning habitat, or only a few males fertilizing eggs.
So that's an interesting question. And one of the things that we've been able to do in populations across the coast is use our genetic data to reconstruct the parentage of cohorts of juveniles and essentially see how many males and females contributed to each cohort. I don't have information in front of me for the Hudson River population, but in general, we haven't seen any really strong signs of kind of a sweepstakes reproduction where a few animals really drive things. But it is an interesting question. Like, you would think that the males and females would spend more time together, and I don't have a really good answer for you there.
Thanks. George Jackman, he asked, how is it that adult non-natal males can find or know where Hyde Park spawning grounds are located? What makes them travel up to a non-natal river, presumably to spawn?
Sure, that's another one that we're trying to sort out. I don't have a concrete answer for you, but I can share a few thoughts. So one is, we don't know for sure that when they're upriver that they are actually trying to spawn. Sturgeon live a long time, they're very mobile. It could just be that in their wanders, they've ended up there. It could also be that they are cueing into some sort of cues, either following other fish or detecting pheromones of some sort that are interesting to them, that encourage them to move upstream, but then they get there and they don't actually attempt to spawn. There's also an interesting body of work from trout and salmon looking at female choice in fishes, which historically was overlooked, but it turns out, at least in some species, females have a lot of ability to ensure that their eggs are fertilized by partners that are likely to promote fitness. And one of the ways that works in salmonids is the ovarian fluids actually stimulate the sperm of the correct partners differentially when compared to poorly suited partners. We don't have any evidence that that's happening in sturgeon one way or the other, but that is one mechanism where that limits hybridization or integration in other fishes. The last thing I'll mention is there's a theory for migration in fishes called adopted migration, and that's basically suggesting that one of the ways that fish learn their migration paths is essentially through social cues, where we know that sturgeon form these mixed stock groups out in the ocean, and maybe they just kind of tag along with the group. We're really not sure that's something we want to dive into a little further.
Right, thanks. We have a question that, given that that sturgeon are benthic feeders and spawners, can you talk a little bit about why we still have vessel strikes, and is that related to, you know, their migration patterns or other activities?
Sure. So that is accurate that sturgeon spend most of their time near the bottom. But it's also true that vessel strikes are a big problem, at least in some systems. The Delaware River is one, I believe the Savannah River is another, maybe the James to a lesser extent. But the systems where we're seeing a lot of ship strikes tend to be ones where we have sturgeon that are packed into a narrow channel. In many cases, we've artificially dredged that channel to be deeper and more defined, essentially concentrating our sturgeon right where the vessels are going back and forth. And even though the sturgeon are spending a lot of the time near the bottom, in rivers like the Delaware River, the water might be 45 feet deep, but those large ocean-going cargo vessels are sometimes drafting 40 feet. And that only leaves a little bit of space below the vessel. There's also a giant propeller that likely has some serious forces around it that are not fish friendly. So there's, even though it's deep water, there's actually not that much space between the bottom of some of these big boats and the fish themselves.
Maybe sticking on that sort of anthropogenic impacts, there's a question around there's a potential, I think you're aware probably, there's this potential for a storm surge barrier across the either the Verrazano Narrows or Sandy Hook Rockaway transect, and whether you can sort of talk a little bit about what the potential impacts of a barrier like that might be for sturgeon.
That's not one that I'm
I'll give
very
you
well
easy
equipped
ones.
to answer. Yeah, I'm not familiar with that. Shannon, feel free to jump in if you know more.
Okay.
The one thing I will make a comment though, at least,
Yeah.
you know, sturgeon have been around for tens of millions of years. They're pretty well dialed into what has worked in the past to be a sturgeon. I should clarify that they're adapted to past conditions. There's relatively limited spawning habitat in the Hudson River, and they're programmed to be there at a specific time of year. So if access to that is limited, yeah, that could be a big problem.
Right. So yeah, just maybe building on some of the habitat used by sturgeon. You know, there's a lot of people thinking about what we can do to improve habitat for sturgeon and other fish. Can you sort of talk a little bit about what you might recommend? Are there rocks or other materials that might enhance restoration areas? What for sturgeons in particular?
So sturgeons as a group like to spawn over rocky substrates. And across the range of Atlantic sturgeon, they're able to find those habitats in different places. The Hudson River, we've got a decent sized rocky outcrop near Hyde Park that seems to be, based on telemetry data and some other observations, probably the epicenter of spawning, with several other satellite sites that are potentially of use. I personally haven't seen anything to indicate that spawning habitat is currently a limiting factor in the Hudson River. It may be in other systems, and I'll mention that folks who are responsible for managing lake sturgeon in the Great Lakes have, in some cases, created artificial reefs of rocks that are now supporting sturgeon spawning. So there is a precedent with this group of fishes. I'm just not sure it's necessary for the Hudson River. Because sturgeon are such, take so long to reach adulthood, I'm more concerned personally, like if I was trying to figure out where we have the most opportunity for leverage, I think reducing adult mortality is probably where I would focus.
You mentioned in the work at the Great Lakes, there's a question around how the Atlantic sturgeon differ from those found in the Great Lakes and in other areas, I guess.
So yeah, so obviously they use salt water, but one big difference is they're able to migrate over huge areas, whereas lake sturgeon they do still move around seasonally, but they're confined to much smaller spatial scales.
So actually, I have my own question here. Are you working in any of the offshore wind areas? And I mean, you mentioned that there's a bunch of offshore telemetry work going on. I guess the question is, do you see that as an opportunity, given all that potential new work and research being done to get a better handle on any aspect of sturgeon ecology, I guess?
Sure. So as most people on the call are probably aware, offshore wind has gotten a ton of interest in over the last decade or so, and projections are that we will have thousands of large industrial scale offshore wind turbines on the continental shelf in the next few decades. That is what I would consider to be a new threat. It's associated with both like pile driving and ship traffic, and also some undersea transmission cables, all of which are new dynamics for sturgeon. And in association with that interest in offshore wind, there's been a fair bit of funding that's been opened up for sturgeon research in those offshore areas, because in the past, the ocean was just kind of this black box where we knew these fish went, but we knew little about their activities out there. And we also kind of assumed naively that most of them were spending most of their time close to the coast. More recently, telemetry data suggests that, especially in winter, these fish actually spend a lot of time pretty far out on the continental shelf. So we're looking at this wind energy question in a couple ways. Shannon presented a slide on this large collaborative sturgeon telemetry project that we're leading. And the idea in that project is to better understand when sturgeon are present in offshore wind areas and which populations are there. The which populations becomes important under a Endangered Species Act management context. And then we're also, as Shannon mentioned, starting a project, be going out in the field in August, to satellite tag 25 Atlantic sturgeon and see when and where are those fish in these offshore areas. It's a big ocean, and so far we've only been able to detect fish when they swim within a few hundred meters of a receiver. And also trying to use side scan sonar in some offshore sand areas. Actually the focus is on sand mining for things like beach renourishment, but we're in an area that's very close to some offshore wind proposed lease areas. All of which trying to get a better idea of when and where are these fish out in the ocean, what are they doing, what habitats are important that we might need to protect.
That's great, thank you. So we had a couple of early questions around study design. Jeff Leamington asks what the age range of the juvenile surveyed for the natal origin within Haverstraw Bay were. So age range of the juveniles.
Shannon, can you take that one? Yeah. The total range was, I think, 1 to 11 years old, but the overwhelming majority of fish that were caught in that survey were between 3 and 5 years old. So I think it was over 60 or 70% were between 3 and 5 years.
Thanks. So another sort of study design, Fred Jacobs' question. Given that only a percentage of the spawning adults return to the river to spawn each year, what is the best estimate for the total adult population size?
Yeah, so I don't have a hard number on that one, but I can give you a back of the envelope estimate. So we know from more recent work, including some focused on the Hudson River by Matt Reese and colleagues, that males are returning to the Hudson River, most years, not every year, but close to annually. And females in the Hudson River system average, I think, just over a 2-year spawning interval. So if we kind of aggregate those, again, back to the envelope, the total spawning population size should be roughly 1.5 times what we see in a given year. Our 2014 estimate was 466 animals. that puts us probably in the 700-ish total fish, at least at that point in time.
700 total in the.
Yeah, in that ballpark. I mean, there's confidence intervals on our estimate, and doing some rough estimates to get there, but it's still not that many total fish, and we don't have to look that far back in the historical records to see similar numbers of fish being harvested in a single year.
Sure. And how many have tags?
From the Hudson River, a few hundred total fish currently.
Okay, well, that's interesting. So another question. Let me see. I think you suggested that you didn't expect non-natal individuals to spawn, but the question is, might they? And then if they do, can you develop some sort of mixing rate?
So from our genetics data, we don't really see any indication of focus on the Hudson River, but this is true across the range. We don't see much evidence of hybridization between, or intercrossing, whatever we want to call it, between populations. And there's a lot of steps that go between a fish entering the wrong river and being able to see hybridization. It has to go to the right time and place. It has to find a receptive partner. The eggs have to actually be fertilized, and there could be mechanisms that prevent that. I mentioned some of the female choice theories earlier. And then if there is fertilization, those fertilized eggs have to survive and grow and make it all the way up to a size where we're able to sample them. And there's a lot of links in that chain where things could not work out.
So maybe we have one or two more. So on the offshore wind, Marcia Johnson is asking whether, given their, the structured habitat being added as part of those wind turbines, and that there's some amount of protection potentially from boats, approaching those structures, could they in fact be a refuge for some species?
So certainly, adding new structure offshore is going to benefit some species. I would guess things like black sea bass probably might really like that. For sturgeon, to be completely honest, I think we should do due diligence and figure out the potential risk vectors to sturgeon with offshore wind. But to be completely honest, my professional opinion is that some of the other existing risks are probably of much greater consequence. I think there'll be likely some short-term impacts due to pile driving, and there is a risk of some vessel strikes. But if we take a panoramic view, I think we should take due diligence when it comes to introducing a new threat like offshore wind. But I'm personally not convinced that that's going to be a game changer relative to the other threats that they face.
So just follow-up question on potential impacts. George Jackman is questioning whether are sturgeon sensitive to EMF from the power transmission cables.
I think that's an interesting question. I believe one of our colleagues, or perhaps several of our colleagues, are working specifically on that power transmission line, but I can't comment on that. I don't know one way or the other.
Thanks. I'm not sure what this is, but Richard McBride wants to know if you can estimate Hudson River population using a close-kin mark recapture method.
So the close-kin mark recapture methods are kind of a new set of models that leverage high-resolution genetic or genomic data to estimate abundance. And basically you're looking at multiple generations and reconstructing more or less family trees of sturgeon. We've thought about it. The microsatellite panel that we currently use for most of our work probably doesn't have quite enough resolution. Sturgeon are also pretty tricky to use that model on because they live so long and have so many overlapping generations. My understanding that the Scribner lab out of Michigan has tried it for lake sturgeon, and it's been difficult even in systems where we have a really good idea of what's going on and a lot of samples.
Maybe the last one. There's a question about how you age sturgeon and whether you can count the rings in a cross section of I'm not a fisheries person, of a skute. I'm not sure what that is. Or are there bony spines in the fins? Which is just a technique, I guess, question about how you age sturgeon.
Bony spines in the fins are probably the primary way that people are exploring currently to age sturgeon. That's likely to be more effective on smaller fish than older fish. As they get older, the rings probably get really close together, and there's just a lot of them to count. The Atlantic States Marine Fisheries Commission is currently leading an effort to help standardize aging across different agencies using fin spines and fin rays. Not sure where that is currently, but it is underway. I actually think it would be really interesting to use something like bomb carbon isotopes to try and age some of these really large fish. And my suspicion is, we mentioned a maximum age of about 60 years in the literature. My suspicion is that some of these fish are probably actually quite a bit older than that. In lake sturgeon, we know that those can live well over 100 years. And I just don't think that our hard part counts are probably capturing the full potential longevity in this species.
Well, Dave and Shannon, thank you again. As you can tell from all the comments, I think people really enjoyed your talk, and we all learned a lot. So thank you so much. And thanks to everyone for joining. Have a good day. Thanks, guys.